Good morning everyone. So for today's class we're gonna continue with the cell division for the last part. Okay, now move on to the next slide. So we're going to the first point here is the importance of control mitosis. Okay, control mitosis, which means that the process of mitosis is under control. Okay, control mitosis is the ability of the cell to divide at its own rate and time. So we can see that is the meaning of that the mitosis process is acting in normal way. So it's important, crucial to normal growth, development, and maintenance for the perpetuity of living things. For example, in plants, control mitosis happens at shoots. On the shoots result in growth of plants. For in humans, Malpighian layer of skin divides actively to replace dead and damaged cells. Alright, so the Malpighian layer is a layer in the epidermis in which skin cells are continually formed by the division. Okay, let's see some example here. Epidermis, where you have one of the layer called as a Malpighian layer. So it's a layer of cells which produce the pigment of melanin. Melanin gives the skin and hair its color. If you look this diagram, so this is the hair on the skin. So this is the skin here. So the melanin protect us from ultraviolet rays, which is coming from the sunlight. So the arrow pointed here show you that the Malpighian layer. Next one. So the effect of uncontrolled mitosis in living things. Right, so previously you learned is about the controlled mitosis. How about when you have uncontrolled? Which means the process is uncontrolled. So what's the effect due to the human body? So the first thing you should know is the mutation. The term here, mutation, is a change in the DNA structure of the cell. DNA structure, as you learn that, the function of DNA is to store the genetic information. So let's say the information has been replaced or missing. So this can disturb the process of mitosis. The process that been disturbed is caused by the mutation. So the change in the DNA corrupts the coded genetic instruction for mitosis control. Right, so when that happens, so it can lead to uncontrolled mitosis, which is the non-stop division of cells, producing a mass of new daughter cell, which is called tumor. So uncontrolled mitosis, here you have two diagrams here. This is the normal one. If you compare to this one, malignant. Malignant. What is malignant? So this one here is say cells are cancerous and can spread to other tissues and organs. So we can say that malignant is a stage in our body where can be very dangerous to the human's health. Malignant, as it say here, malignants are cells that cancerous. Cancer, so which can give a very bad effect to the human's body. So now we go to the what is cancer. Cancer is uncontrolled and abnormal. The keyword here, the capitalized words. Cell growth that can spread to other parts of the body, disrupting normal bodily function. So that means that the cells in your body grow too quickly. When this occurs, some of the cells stop working properly. Sometimes cancer cells grow so quickly that they may cause a lump or tumor to form somewhere in your body. If we compare to these two diagrams, so this is the normal cells. If you compare to this one, you have this dark yellow star shape. Right, so this is the cancer cells. Cancer cells grow uncontrollably. It will crowding out the normal cells. Which means that disturb the normal function of the cells. Right, so that's the cancer. Cancer from tumors. Tumors means masses. That deprive other healthy cells of nutrients. So you have one process here known as a metastasis. So when cancer cells break free of original tumor and spread via the circulatory system to other parts. Okay, this leads to new tumors. Okay, metastasis means that where you have this one tumor cells attachment sites. Okay, initially this is where the tumor cells grow. So when it break apart. So it will go to other parts of the body. Okay, you can see that the movement here and go to other parts of the body. So we say this is a secondary tumor. Okay, so this is the process of metastasis, which means that the tumor growing apart from the original location. Okay. Okay, so causes of cancer: genetic, okay, carcinogens, radiation, and virus. Okay, genetic, okay, some form of cancer like prostate, colon, breast, skin, ovary are suspected to be inherited from the parents. Okay, yeah, carried by generation to generation. What it means by this uh, first point here. So carcinogens, okay, is the chemicals, okay, which affect the genetic activity and cause cancer. For example, like carcinogen, diesel exhaust, okay, cigarette smoke, okay, dyes, shoot, okay, which is the carbon, arsenic, benzene, and formaldehyde. Okay, another one is the radiation, okay, coming from the exposure, okay, excess exposure to X-ray, gamma rays, and UV rays, okay, will lead to increase in cancer risk. Okay, some virus, okay, such as the HPV and HIV-1 can cause cancer. So all of these four factors are the causes of cancer. Okay, so now we go to the next one, application of mitosis in cloning. Okay, so mitosis is used to improve the quality, to produce new species, and to ensure uniformity in the traits. Okay, as you know that the mitosis carry the same traits. Okay, cloning. What is cloning? So it's process of artificially creating a new individual that is genetically identical to an existing individual. Okay, technique in application of mitosis. Okay, in cloning, normal one is the tissue culture. Okay, so application of mitosis in cloning. So this is the example here. You have the plant tissue, so you have the skin tissue. Okay, and then you have here tissue is removed, okay, from the duodenum, okay, which is a small intestine, okay, to make a culture for analysis. Okay, so this is application of mitosis in cloning. Okay, now we're going to the next one. Okay, how meiosis, okay, previously is mitosis, now we go to meiosis. Okay, how meiosis provides for genetic variation. Okay, we have different why we have person have different color of hair, different color of eyes, okay, somehow like that. Okay, so why we don't all look alike? Okay, so we have the crossing over. If you still remember the, the step, okay, in prophase. Okay, so crossing over, this is the main reason. Okay, rearrange genetic info. Gene from each parents don't stay together. So which means that moms get mixed with dads. Okay, the genetic information. Okay, and also chromosome don't always get split the same way. Okay, they line up in different ways, so each sperm or egg has a different combo of genes. Okay, so these are the two main reasons why we all don't look alike. Okay, so this is the diagram showing that the crossing over, this one. Okay, and then at the end you have the genetic information is exchanged. Right? You can see that no, let's just not in the same color anymore. Okay. Okay, so when meiosis goes wrong, so what will happen? Okay, so we review back. So you know that meiosis is used to make gametes. Okay, only gametes. So when it works, okay, and usually does, okay, each gametes end up with haploid number of cells 23 for humans. Okay, 23, 23, 23, 23 here. Okay, okay, so meiosis errors. Okay, but sometimes it doesn't go according to the plan. Okay, mistake happen. So there are two main types of error. Okay, error caused by change in chromosome structure, and the second one, error caused by changes in chromosome number. So let's see what is happening here. Okay, so the first one, error in chromosome structure. So usually occur during the process of crossing over. Sometimes when part of the chromosome get detached and switch places, then small pieces get removed or inverted. Okay, which means put in upside down or switch to a completely different chromosome. Yeah, sometimes only one chromosome lose a piece and another chromosome gets one. Okay, so that's the what is happening during the crossing over. This is normally happens during the crossing over. Okay, where the information can get removed or inverted or replaced somehow like that. Okay, error in structure, okay, chromosome structure. So when any of these things happen, there is a change, okay, that the baby that forms from one of the gametes will be deformed. So you have this one, chromosome mutation. Okay, this the okay, we will learn this in the next chapter. Okay, but this is the process, okay, where you can lead to error in the structure, chromosome structure. So the first one will be deletion, okay, inversion. You have duplication, okay. You can see the duplicate, the mistake here, and then translocation, okay, where the information is translocate translocate to different position. Okay, inversion, which mean okay upside down, okay, inverted. Okay, deletion, which mean remove. Okay. Okay, so structural disorder can lead to many abnormalities and increase the likelihood of some disease, okay, including cancer. Okay, so this can cause, okay, in chromosome structure can cause cancer. Cancer of the Cradut syndrome is a deletion disorder where you have the information has been deleted. As you can see here, if you compare here, you can see at the end here you have the part that has already been deleted. Okay, we name this as a Cradut syndrome. Okay, this is a diagram showing the person with Cradut syndrome. Okay, errors in chromosome number. That's all. Okay, so it occurs when homologous chromosome or sister chromatids don't separate properly in anaphase 1 or 2 stage. So this is called non-disjunction. Here you can see that it's a normal, and then, okay, let's see the normal first part here. Okay, you have the meiosis. Okay, you remember, right? You have meiosis 1, meiosis 2. So this normal, so when it divides, so it will undergo, okay, this one undergo normal. So this one, if you see here, take note on this place, okay, on this part, okay, on this part, you can see that non-disjunction. So you can see here you don't have any chromosome. This one you have, okay, which means extra one. Okay, another one here, another example here. When this normal cell undergo non-disjunction, okay, so you have this one. This we have no nothing here and nothing. So this one you get extra, something like that. Okay, we call this as a non-disjunction. Okay, non-disjunction occur when problems with the meiotic spindle cause errors in daughter cells. You can see that, right? This one non-disjunction. So if you will go extra here, you will have extra chromosome here. Again, this one, same goes to this one. Supposedly they have to break into this one equally, but now you have here you have extra one. Okay, now you okay extra one, so this one you have minus one, something like that. Okay, it must be equally. Okay, so this may equal if the tetrad chromosome do not separately properly during meiosis 1. Alternatively, sister chromatid may fail to separate during meiosis 2. Okay. Okay, now we go to the next one. Okay, when non-disjunction occur, one gamete will lose a chromosome and one will get extra one. Okay, an extra one. So both cases can cause defect. Okay, as you can see from the karyotype chart, this is the karyotype chart. Okay, karyotype chart. So the later chromosomes are shorter. Okay, since they are shorter, they are more likely to get moved to the wrong place. Okay, so this is showing you the karyotype, uh, karyotype chart for okay for those people that have a errors in chromosome number. Okay, you can see this one. Okay, you can see that you have the chromosome here, you don't have Y chromosome here. Okay, chromosome here, you get extra here. Okay, it must be equally, okay? So it must be something wrong, okay, at this, okay, on this part. Okay, non-disjunction disorder, the first one will be trisomy 2, okay, which is known as Down syndrome. Okay, cause the 3 chromosome, okay, 21. 3 chromosome 21, okay, which basically means that, okay, for those person who have Down syndrome, on the 21 they will have 3 chromosome, okay, somehow something like that. Okay, changes 1 to in n100 births, okay, ratio. Okay, more common with older mothers. Okay, the sign is flattened face, okay, intellectual disability, okay, cannot speak properly, okay, and then short stature, okay, the body structure. Okay, another one will be trisomy 18, Edward syndrome, which is known as, okay, trisomy 18, okay, cause 3 chromosome 18. Okay, if you go back to the karyotype chart, 18 here you have extra one, okay, something like that. Okay, the sign here, facial abnormalities, okay, intellectual and physical disabilities. Okay, so this diagram showing the person is having Edwards syndrome. Okay, non-disjunction of sex chromosome. Okay, normally you have the Klinefelter syndrome. Okay, where you have extra one, okay, extra X chromosome. Okay, so the signs here, you have taller, less muscular body than males, their, okay, their age, it is their, okay, THEIR, broader hips and longer legs, okay, larger breasts, cubical bones, a lower energy level, smaller penis, okay, and testicle, delay in puberty or grow a partial amount, okay, less facial and body hair following puberty. It is also are the signs for the person that having Klinefelter syndrome, okay, where the person have extra X chromosome. Okay, the second one will be the Turner syndrome. Okay, sex chromosome where you have only X chromosome. Okay, you don't have Y. It must be have both, okay, X and Y. But this one X or means the person doesn't have any the type of chromosome. Okay, so it only have one chromosome. So it's only normally happen in females. Okay, Turner syndrome. Okay, 1 in 5,000 female ratio. So this is the sign. Okay, you have the short stature, body structure, short. Okay, webbed neck. Okay, you can see this neck, and you have a sexual immaturity. Alright, so there's a Turner syndrome. Okay, signs. Okay, so that's the end for this chapter cell division. So thank you everyone for listening this video. Okay, and watching this video also. Okay, thank you.